A very good afternoon to one and all present here. I, Nikhil B. Mohan, on behalf of the entire School of Law of Christ deemed to be University Lavasa, welcome you to the third session of the St. Kuriakose Elias Chavara lecture series. Today, I extend my heartiest welcome to our chief guest and keynote speaker, Dr. Susan Eapen, a former scientist from the Bhabha Atomic Research Center in Mumbai, to our respected director, Reverend Father Josie P. George, our honorable HOD, Ms. Daisy Alexander, our esteemed faculty members, and all my friends. January 3rd is celebrated as Founders Day in reverence to our founder, Saint Kuriakose Elias Chavara. Saint Kuriakose Elias Chavara was a devout Christian clergy who did what many people together cannot achieve even during the course of many centuries. He led an extraordinary life of holiness and committed action for the transformation of society. Saint Chavara was a visionary leader of the time who envisioned many plans for the spiritual and social progress of the church and of society. His educational contributions laid a strong foundation for the just society in which all human beings were worthy of erudition, justice, respect, and dignity. Saint Chavara was a self made social contrary to the common practice at the time. He was a creator for the ages. As a gesture of reverence and to inspire the present generation of young adults and to discover the greatness of Saint Chavara, we at School of Law, Christ deemed to be University Lavasa, have initiated this law lecture series. The objective of the series being to invite leaders from different walks of life who have contributed to the transformation of the society around us. Now, without further ado, I request our our HOD, Professor Daisy ma'am, to kindly welcome the chief guest and the keynote speaker.
Good afternoon, everybody. This is a third lecture by Dr. Susan Eapen, a former scientist from the Bhabha Atomics Research Center. We welcome you, ma'am, and I would request our director, Reverend Father Dr. Josie, for the opening remarks.
Good afternoon, everyone. We started with this lecture series 2 days ago. And I hope that I speak for all of us when I say that the first 2 days have been an informative and eye-opening experience for each one of us. The lecture series initiated to commemorate our founder, St. Kuriakose Elias Chavara, has so far been truly engaging and inspiring. To continue forth with this journey of inspiration, we have with us today Dr. Susan Eapen, speaking on the topic of much relevance to the time living. Maybe you will be getting the official notification within 2 days that we are going to open the campus for offline classes from February 1st. So as we are preparing for this coming back to the campus, I think such a lecture series will be very much helpful for our life. And beyond just the role of students, all of you must rise up to be the socially conscious individual. And I hope Dr. Susan's talk will guide you in the right direction. Once again, I appreciate the School of Law for taking this such a great initiative. I'm sure that this will continue every year, this lecture series. Without much delay, let me conclude my words. I hope the lecture series will continue to enlighten and enrich young minds to deliberation on the relevant social issues and act as responsible individuals working for the collective good for the society. So thank you very much and all the best.
Thank you, Father, for those words. Now, I request our Student Bar Association member, Nishant Venugopal, to kindly introduce our chief guest and keynote speaker.
Dr. Susan Eapen is one of India's preeminent scientists and one of the pioneers of transgenic plant research. Stanford University recently included Dr. Eapen in the world's 2% top-ranking scientists in the category Plant Biology and Botany. She was the president of the Indian Women Scientists Association. During an Indo-FRG bilateral program in 1986 at Free University Berlin, she developed the first transgenic grain legume from protoplast using 3 different techniques. Later, she developed transgenic plants which could take up heavy metals and degrade organic pollutants. She was also involved in the Cotton Mini Mission project of Government of India and was a collaborator for several BRNS funded programs all over India. Scientists from Russia, Cuba, and Syria have come to her lab to get trained in latest techniques. She had been in the editorial board of international journals In Vitro Plant and BMC Biotechnology. She received Elsevier Award in 2010 for the Best Quoted Paper of the Year. She is also a recipient of UNESCO and British Council fellowships. A 15th batch BARC training school scientist in biology and radiobiology, she retired in the year 2011. Currently, she is engaged in motivating scientific temper among faculty and students at the Union Christian College, Adwa, as an adjunct professor in biosciences group. Her interests varies from plant biotechnology, production of secondary metabolites from in vitro cultures, and phytoremediation. She has published 140 research papers, contributed several book chapters, and presented papers in several national and international symposia. Her latest paper is on phytoextraction of uranium using material plants. On behalf of the entire School of Law, Christ Deemed to be University, Lavasa, Pune, I thank you for accepting my invitation to deliver this lecture. Thank you, ma'am.
Thank you, Nishan. Now, I am honored to welcome our keynote speaker, Dr. Susan Eapen, to speak on the topic Plant for Feeding the World and Cleaning Up for Water Bodies.
Hello, can you hear me? Hello, can you hear me?
Yes, ma'am, we can hear you.
Yeah, I'll start my presentation. Good evening, everybody. Thank you, Dr. Daisy Alexander, for inviting me to deliver this lecture. Father Josie George, Mehak Ora, Sunil John, and all the others. I am actually speaking for lay people so that you understand the subject. So today's talk is on plants for feeding the world and cleaning up of water bodies. Before I go to the actual topic, I know that you are from different fields, so I will speak a few words on journey of biotechnology. What is biotechnology and what has happened in the 19th century and the 20th century, and what is happening at the moment. Biotechnology, as you know, has been practiced from time to time, from a long time onwards. As you know, that cheese, wine, curd, etc. are prepared from, for example, wine is prepared from grapes from time immemorial. But we did not use the word biotechnology. Actually, the word biotechnology, the term was coined by a Hungarian engineer, Karl Erecki, in 1990 when he developed bioproducts using biological organisms quite quickly and rapidly. Currently, biotechnology is used to improve human health and also environment and to produce novel plants. We can see here a tomato which can be left for a long time without decay. So you can produce vaccines, novel pharmaceuticals, novel foods, agricultural products, etc., to improve our life. So this is biotechnology. You can clone a gene from any organism and introduce into some other organism, produce medicines, etc. Let me go back to the 19th century. In 19th century, maybe you would have studied in your school that a monk, a priest, Gregor Mendel, lived in a place called Brunn near Austria, and he developed the Mendel's laws of inheritance. He was actually basically a mathematics person with a lot of good information on statistics. He was very much interested in inheritance, how characters from parents transferred to the offsprings or to the children. For example, he was always finding that dogs always give birth to puppies and cats always give rise to kitten, and human beings give rise to human babies. How does this happen? How does the children resemble their parents and their grandparents? He was very much interested in this topic, and he started studying, doing experiments using mice in his monastery. But other priests objected to keeping mice in the monastery. So he started using pea plants, garden pea. So he selected garden pea plants from different areas, and he selected plants which are having different characters. For example, tall plant, dwarf plant, flowers having pink color, white color, then full seed, shrunken seeds, yellow seed, green seed, etc. For he developed he collected many plants from different areas and self them, inbred them, and maintained in his laboratory. So this is what the experiment he did. He took a pea plant with purple flowers. Sorry.
Microphone on nahi hai.
With a purple flower and crossed it with a white flowered pea plant. He crossed a white flowered pea plant with a purple flowered pea plant. And to his surprise, all the offsprings in the first generation, they were all purple. Then he wondered, what happened to the white color? Has it disappeared? What happened? Then he went to the second generation. Then he found that there were purple flowers, also white flowers. And when he calculated them, there were three in the ratio of 3 purple flowers and 1 white flower. So that means the white character, although did not express in the first offspring, it had come to the second generation. How did it come? So he did statistics. Also, he studied different characters. He took a tall plant and crossed with a dwarf plant. He took a plant with the flowers on the axils and he crossed it with the plants with the flowers on the top, then with a yellow seed and the green seed, then a shrunken seed and full seed, different coat color, etc. So 7 characters he studied in the pea plant, and he reached the conclusion using statistical analysis that characters, for example, color of the flower or seed shape or height of the plant or the place where flowers are produced, like a top or the axil, all these are characters. These physical characters are inherited as particles. He did not know anything about DNA. He did not coin the name gene. He just said characters are inherited as particles. And they are passed on from parents to offsprings. And the traits remain as alleles. That means there are 2 for each character. For a purple color, there are 2 alternatives, 2 alleles. They may be of same allele or different allele. So one allele can be dominant over the other. That is, purple color can be dominant over white flower. That is why in the first generation, purple color is expressing. So based on his studies, he published his work. And but his work in the 1965 went unnoticed. Nobody noticed his work. And he passed away without even getting any recognition. So during that time, there was another milestone in biology. You all are familiar with Charles Darwin, came with the theory of evolution and how man came from ape, etc. So he said, it is not the strongest of the species which survive, but nor the most intelligent, but the one most responsive to change. So one has to change with the time, like how we are changing with during the lockdown, how we are changing like by taking new options, like like this webinar, etc. The animals and the other plants have to survive. If they have to survive, they have to change with the time. It is not your intelligence that will help you to survive. For example, suppose there are many butterflies and moths. Butterflies are very colorful, and if a predator is coming to eat catch the butterflies, they will prefer to catch, they will easily catch the colored butterflies. And the moth, which is very much resembling the dry leaves, will escape. So Charles Darwin came with his theory, and Charles Darwin was not knowing anything about Mendel because there was no internet or Facebook or anything during that time, and all the work of Mendel went unnoticed. And during that time, there was another great scientist called, you all have heard about Louis Pasteur. He was a French chemist and a microbiologist. And at that time, there were so many diseases prevalent among people and animals. And he showed that microorganisms or germs cause diseases. Diseases are caused by microbes or microorganisms. He developed a vaccine actually for anthrax. And at that time, rabies was a very dangerous disease, a dangerous problem, because if a dog having rabies, if he bites you, he will definitely get rabies and you will die of that. So he developed vaccine for rabies. Then he also developed a process called pasteurization, where you can heat, for example, milk is heated to a particular temperature and cool down, and the process is known as pasteurization. These are the milestones in biotechnology in the 19th century. Then we come to some other milestones in biotechnology. You know that cells in your body, they always divide. But when they divide, they have a particular number of chromosomes, and that is maintained. That is known as mitosis. Cell division in the somatic cell is known as mitosis, because after mitosis, there will be the same number of chromosomes in the daughter cells. And in 1873, microorganisms were stained by Robert Koch to distinguish different microorganisms, like a gram staining was used. Then in 1900, after Mendel passed away, Mendel's laws were rediscovered by 3 groups of scientists from across the world. Then only they came to know the importance of Mendel's laws of inheritance. But Mendel did not know anything about gene. He did not know anything about chromosomes. It was only in 1902 that Walter Sutton said that chromosomes occur in pairs. So what Mendel had postulated, that characters remain as alleles, as 2, came true, because chromosomes remain as pairs. One set of chromosomes come from your father and one set of chromosomes come from your mother. That is why chromosomes pair and remain like that. So then came T.H. Morgan. He worked on fruit fly. When you keep fruits on your table and when they get very ripe, you can see a lot of small flies coming and covering the fruit. They are the fruit flies or Drosophila. So Morgan designed a large number of experiments on fruit fly with red color, then white eye color, etc., and the sex-linked chromosomes and everything. And he derived a lot of observations and all based on his work in fruit fly or Drosophila. Then came William Johansen, and he is the one who actually called the name gene. Up to now, Mendel was speaking about characters. Nobody called gene. He is the first one who coined the name gene. Then I would like to tell about some milestones in biotechnology. For example, Frederick Griffith. Nobody was knowing about DNA at that time. He was the first one who said that there is a transforming principle. How did he reach that conclusion? He was working on bacteria and a virulent strain, which is shown in red color, and a non-virulent strain, which is shown as blue color. When he infected non-virulent strain into mice, what happens? The mice will survive. Nothing will happen to the mice. They survive nicely. Whereas when you inject a virulent strain into the mice will immediately die. Then he thought, let me do a third experiment. In the third experiment, he took the virulent strain and killed it with heat and then injected into the mice. Surprisingly, all the mice survived. That means when the bacteria is killed by heat, something is inactivated and it is not transferred to the mice when it is being injected, and the mice survives. Then he did a fourth experiment. He took the heat-killed virulent strain, and he took the live non-virulent strain and mixed both. Mixed the live non-virulent strain and heat killed the virulent strain and injected into the mice, and all the mice died. That means if something is moving from the virulent strain into the non-virulent strain, making it virulent. And what is that? He called it transforming principle. Then later in 1928, you all are familiar with Alexander Fleming and his discovery of penicillin, which was very helpful to soldiers of the World War. And now a lot of penicillin, a lot of antibiotics are available, like cefotaxime, etc. Now in 1941, Beadle and Tatum said that there is one gene will give rise to one protein, and he came with he worked in Neurospora crassa, which is a fungus. In 1945, for the first time, animal cells were grown in vitro. During the same time, plant cells were also grown in vitro. But there's a difference between animal cells and plant cells. As far as plant cells are concerned, they are totipotent. What is totipotent? Totipotent means that if you take a cell from the somatic cell and culture them, it will have a potential to become an embryo and a complete plant. But animal cells lack the potential to produce a complete animal from a somatic cell or a cell taken from the animal's body. So plants are totipotent. So each cell in the plant, when you are taking from a tissue, can become an embryo and become a complete plant. This was demonstrated first time in carrot. In carrot, root cells were cultured, and they were when they're cultured, they produce a callus. And when they are callus, from the callus, you can remove some of the hormones and put it into shake flask. And then each cell will transform into an embryo. So millions of embryos are developed from a few cells, and they will produce a complete plant. So plant cells have the potential to have for a totipotency to produce a complete plant starting from a single cell, whereas animal cells did not have the potential for totipotency. Now comes 1953. As you know, you all have heard about DNA. The structure of DNA was elucidated by Watson and Crick, and they got Nobel Prize in the year 1962. The DNA is a the double helix. It has got a, it has a helix that here you'll have a deoxyribose as a sugar and also phosphoric acid, and the steps are made out of certain guanine and cytosine, adenine and thymine, like that. So this is the structure of DNA elucidated by Watson and Crick in 1953, and they got the award. So later in 1956, Severo Ochoa cracked the genetic code, like 3 letters of the DNA, that is adenine, thymine, guanine, and cytosine. 4 letters are there. Out of 3 will at a time will code for a amino acid that is known as a genetic code. This was cracked by Severo Ochoa, and his student Arthur Kornberg synthesized the infectious viral DNA from and for the first time, and both of them got the Nobel Prize. I met both of them during a meeting at Miami in US, sponsored by the Nature Biotechnology, and I happened to meet both of them. Other milestones in biology, biotechnology are in 1972, Paul Berg found out it is possible to have restriction enzymes to cut DNA, to cut and paste DNA like you want. You can cut the DNA and you can bring it together, paste it together. So these are known as restriction enzymes. So the first time restriction enzymes were discovered in 1972, and this led to gene cloning. There was a revolution in gene cloning with the observation of the discovery of restriction enzymes, and also ligase for fusing the broken strands together. You know about human insulin, that when we have diabetes, when you have a very high blood sugar, many a times we take insulin injection. From where does the insulin come? Normally before 1978 and all that, insulin was collected from the pancreas, collected from the slaughterhouse, from the pancreas of animals like a pig and cows and other animals which were slaughtered. But in 1978, they came that they showed that the human gene can be cloned into a bacteria, and the bacteria can be grown in the laboratory in a flask, and you can isolate human insulin gene. So it became the first commercially produced cloned product which was useful to man. Then came Carrie Muller, who developed the polymerase chain reaction. Now all of you are familiar with RT-PCR. What is RT-PCR? RT-PCR is a advanced version of polymerase PCR. Polymerase chain reaction is how you using a Xerox machine, you multiply, you take a large number of copies. So the copy of DNA can be increased by polymerase chain reaction. And in 1985, you all know about DNA fingerprinting. DNA fingerprinting is used for at a site where certain crimes have taken place, to identify people who have committed the crime, and also for determining the parentage, who is the real father of the child. Sometimes there'll be court cases, and as students of law, you will be using the DNA fingerprinting to identify the actual father of the child. So DNA fingerprinting came into existence in 1985. Also, genetically engineered plants were also produced in 1984 to 85. And for the first time, recombinant vaccines were produced in a plant called tobacco. Then, to extend the life span of tomatoes can be plucked, and you don't have to keep in the fridge. You can keep it outside for 1 or 2 months. To extend the shelf life, antisense RNA was introduced into tomato plants. And the first gene therapy was developed in 1990, because gene therapy, suppose you have inherited a defect from your parents, we can cure it by gene therapy. Then in 1990, people decided that they should the whole human genome should be sequenced. So human genome project was started. They started sequencing the whole human genome. Genome is the whole DNA in the human beings in 1990. And you are familiar with the cloning of Dolly, a sheep. Actually, animal cells, as I said, they are not totipotent, but they induce totipotency. How? They took a ovum, a single egg cell from the sheep, and enucleated it, removed the nucleus of the egg cell, and introduced a somatic nucleus, and they developed Dolly. Then came 1998, embryonic stem cell research. You all are familiar with stem cell research. That human genome project, which started in 1990, was completed in 2003. As a result, 3 billion chemical bases were sequenced, and 20,000 to 25,000 genes were sequenced, and the information is available in the gene bank. As a result of which, the molecular medicine has started. So each of you, like you may be having a different gene sequence compared to mine. So the medicines which will be working for you may not work for me or for another person. So the personalized medicine also started after this era of molecular medicine. So anybody who wants a gene sequence, it is available to doctors and all in the internet. So DNA sequencing was also done in other organisms, like one important bacteria on which people do work in the lab, that is Haemophilus influenza in 1995. The Haemophilus influenza genome was sequenced. Then yeast, that Saccharomyces, was completed. Genome sequence was completed in 1996. And another bacteria which is very, very well grown in the lab, studied, is Escherichia coli. In 1997, that bacteria was genome was sequenced, and there's a worm called Cenobartis elegans, and the work of which had people had got Nobel Prize for working on this. So that genome of that worm or Cenobitis elegans was also sequenced. And the first plant which was sequenced was Arabidopsis thaliana, a distant relative of our Indian mustard or rye or kadu or whatever. Then Drosophila sequence was sequenced. Then in 2013, for the first time, gene editing, CRISPR-Cas9 came into existence. And last year, 2020, two women researchers, Jennifer Doudna and Emmanuel Charpentier, received the Nobel Prize in 2020 for their excellent work on gene editing. It is also called as CRISPR-Cas9. So this is Jennifer Doudna and Emmanuel Charpentier received the Nobel Prize in chemistry for cutting the gene at specific sites and changing it, editing. They are doing editing, like how you do proofreading and editing. Like that, they may found out a new discovery. And now I am coming to the topic of my today's presentation. Is crop improvement for feeding the world. As you know, that world population is increasing alarmingly, and by 2050, we are supposed to reach 9.7 billion people. So you need food security. When there are more number of people, not only we have to provide food for them. So food security is absolutely essential. People should have access to sufficient quantity of affordable, nutritious food. We have to produce more food with less land, with less water, and the climate is changing, and disease and pests and all are affecting the plants. And when the climate is changing, when the temperature is changing, always human beings and can enter their homes, and you can sit nicely in air conditioned rooms, where and also animals and birds can also move into their holes, and they can stay in their tunnels or wherever they want, they can stay. But plants are fixed, they cannot run away from the stress conditions, so they have to adapt to the stress condition. They have to do a lot of chemical reactions. They have to fix their carbon food from the carbon sunlight and from water and carbon dioxide, make food, sufficient food, and take up all the mineral nutrients, do a lot of metabolism and produce what is called as your food. So what are the methods of crop improvement? Crops, always you have to improve your crops to produce more. So one is introduction of plants. What is introduction of plants? Suppose you have a field where you don't get a good yield, and in Australia there is a wheat plant which is yielding very high. We can import that plant. That's a plant introduction, and grow it in India and domesticate it. That is known as introduction of plants. Then one second method of these are conventional methods of improvement: selection. Farmer grows the plant in the field, and the whole field is attacked by insects, but one or two plants survive, and he will select those plants. This is known as selection. Then third type of improvement of plants is hybridization. You can have two parents which are diverse in nature, but from the same species, and you can cross them, and the hybrid plant will yield very high, like a hybrid corn, hybrid tomatoes, etc. So hybridization is another method to bring together genes from a different plants. Then mutation breeding. What is mutation? When a sequence in a DNA changes, a change happens in the phenotype of the plant. That is called mutation. And usually the frequency of mutation is very low in nature, but it can be enhanced by use of radiations or by use of chemical mutagens. So mutation breeding can be used for improving traits in plants. Then latest techniques, that is from 1984 onwards, gene transfer and development of transgenic plants. You can clone a gene from any organism, even from a human being, or from a bacteria, or from a fungus, and introduce into plant to get high yielding plants, to improve the characters of the plants. And latest, now as I said in the last slide, gene editing or CRISPR-Cas9, which is a very precise method of improving plant, not only plants, but other organisms also. Now coming to Green Revolution, yeah, I don't know, you all are maybe, many of you are millennials, maybe others are maybe around 50 or something. So you would not been affected by a food shortage in 1950s and 60s. India had severe food shortage in 1950s and 60s. Everybody said India will be having standing with a begging bowl, and food was imported, like wheat was imported from US under specific programs, and we didn't know what to do, how to, like stand on our own legs. Then at that time, there were some dwarf varieties of wheat in Mexico, which could yield very high levels because they were very dwarf varieties. So at that time, the Professor Norman Borlaug, he was an agronomist, and he also got the World Food Prize. He was in Mexico, and along with Dr. M.S. Swaminathan, who was a director of Indian Agriculture Research Institute, Delhi, and the then Food Minister C. Subramaniam, they decided to import this dwarf variety of wheat and cross with our Indian varieties. When they crossed with our Indian varieties, they developed semi-dwarf varieties having a specific gene called RHT gene, which could stand straight, and it will not fall down. Even when a wind comes, it will not fall down, and the leaves will be very sharp and will be synthesizing their own good amount of photosynthesis, and high yielding wheat varieties were developed. That is how we came across the starvation. And also rice varieties were imported from Indonesia and Philippines and all that. We crossed with our Indian varieties and developed new varieties which could yield very high. So from a begging bowl, the India became a place where we could export food grains to other countries. So radiation induction plants for induction of mutations. When you use radiation, you can increase the frequency of mutations. In Bhabha Atomic Research Center, in my division, we were working on the induction of mutations. So we were irradiating seeds with the gamma rays, and then we were developing the first generation and then the second generation, and we were selecting plants. For example, suppose you have a short plant and you want to have a large number of leaves, vegetative leaves. By induction of mutations, you can develop plants with a large number of leaves, or with disease resistance, or with high yield, whatever. So you have to select and do a lot of work to develop. This way, we have developed 47 crop varieties in my division, that's Nuclear Agriculture and Biotechnology division, and they were distributed after several tests to different, and they're grown in all over the country, right from North India to the South. We have released 15 high-yielding varieties of groundnut, mustard 3 varieties, linseed 1 variety, sunflower 1 variety, cowpea, bigna 1 variety, 2 varieties, mung bean, urad bean. Urad bean is the one which you use for making dosha, idli and all that. Many of the doshas you would have eaten yesterday or today, and the idli that you have eaten, they are from the BRC varieties. Then dal you take, there's a pigeon pea, 5 new varieties were released using mutation breeding. In soybean, 2 varieties, jute and rice, 4 varieties were released. And now they are releasing more varieties using mutation breeding. And now what is and the next technique is genetically modified plants. You have all heard about GM plants. What is the GM plant? A gene can be cloned from a bacteria or from a fungi or from animals or plants to improve the crop productivity, and you can transfer it to your plant of interest. For example, your plant is susceptible to insect pests and plant pathogens. If the moment you grow your plants in the field, lot of insects come and eat them. Lot of virus come and destroy them. Lot of fungi come and destroy plant pathogens. So you can have resistance to all these organisms by transferring gene from an alien source which has got the resistance and develop a transgenic plants. So you can have plants which are resistant to high and low temperature. We can develop plants which are tolerant to flooding, which are tolerant to drought. Then you can improve the shelf life of fruits and vegetables. You can have plants with high yield. We can develop plants which are resistant to pollutants and pesticides. Plants will take up more micronutrients. There's a deficiency in iron, zinc, etc. in our food, and you can improve the micronutrients using the genetically modified technique. So what you do is, suppose you are taking a gene from a bacteria, you clone the gene and take the plant cells, introduce it to it, and then you develop a complete plant. And then they will go into the field. So this is how it is done. Suppose you are taking a gene from bacteria for resistant to insects, you clone the gene from the bacteria. This is the DNA extracted, the gene is cloned, and then you make it into what is called as a vector. Then you take the plant cells and grow it in tissue culture, then grow it in petri dish, introduce the gene into that. From the tissues that are transformed, you select them, develop a complete plant, then transfer to the field, and you test them for disease resistance or insect resistance or whatever trait you are looking for. So this way, we have done, people have developed biotechnology, people have developed the golden rice, which is have got a high amount of beta carotene. Beta carotene, as you know, is a precursor of vitamin A, and the absence of vitamin A, deficiency of vitamin A causes blindness. So in India, this is a very much in need to have this golden rice, but unfortunately, due to politics and other reasons, this is not allowed in India, whereas in our neighboring countries, like even Bangladesh is growing this golden rice, which has got a gene transferred not from a bacteria, from a plant to this rice, so that it has lot of beta carotene. People don't have money to buy carrot and eat, poor people, so they can eat golden rice instead. So this is the first genetically engineered tomato way back in 1984-85. This is called as flavor saver. You can keep the tomato fruits outside in the market or on your table for maybe for 1 month. This is a genetically modified corn, which requires less of pesticides and produce high calories to feed 88 million people more per year. And it is very high yielding, and the income of the farmer will go up by 44.1 billion. Then these are some of the top 10 genetically modified foods like corn, soybean, cottonseed, papaya, rice, rapeseed mustard, then potatoes, tomatoes, seeds, etc. But only in India, they are not grown yet. Now let me go to the development of transgenic plants for metal uptake and xenobiotic degradation. I have written about developing plants for cleaning up of solutions and soil. So a gene is cloned from an alien source and introduced into a vector, and the foreign gene is introduced in the plant by two methods: by direct gene transfer or by agrobacterium mediated gene transfer. In our lab, we have cloned a glutathione transferase gene from a fungus, that is Trichoderma virens, and introduced into tobacco. And we wanted the plant to degrade some of the pollutants like anthracene. A relative anthracene, benzanthracene, is a very complicated carcinogenic compound which is found in the environment, thrown into the environment. It is a polyaromatic hydrocarbon. So we cloned this gene and introduced into our tobacco plants. This is the species of fungus, that is Trichoderma virens, from which we cloned the gene, and then it is introduced into a vector, and then it is used for transformation. And transgenic tobacco plants were developed, and we have to do so many tests, like, this is a transient assay called GUS assay, which is done 48 hours after the experiment. Then so many tests like southern blot hybridization, your RT-PCR, PCR, etc. Then we have to see whether the plant is able to degrade the anthracene, which is a dangerous compound, or not. Anthracene is actually a 3 benzene ring. You can see 3 benzene rings here, and it's a dangerous compound. So this is a control plant. When you grow in the control plant in the soil, the plant does not grow and it almost dies. Whereas a transgenic plant, it grows in the in presence of anthracene supplemented to soil. And we found that tobacco plants expressing this particular gene could take up more anthracene, and anthracene was degraded into smaller derivatives of naphthalene. I'm not going into the details. Then we cloned a human gene, that is cytochrome P452E1 gene. You know, in human liver has got tremendous potential to when we eat drugs, take drugs and all, it can destroy the drugs and cleave it and, you know, like degrade some of the xenobiotic medicines and all what we are taking. So we cloned this very efficient gene from human beings, that is cytochrome P452E1 gene, which expresses in our liver and transferred into tobacco plants. And we tested the plant for degradation of lindane, a pesticide. All these pesticides we have radio labeled and we have studied. So some of the transgenic tobacco lines with a human cytochrome P452E1 gene removed higher levels of lindane from medium supplemented with lindane. So this is what we have seen. Then also we selected some metal transporter genes so that the plants could take up more metals, zinc, copper, nickel, cobalt, etc. Plant can take up. So genes were cloned from Neurospora crassa, which is a fungus bread mold. The transgenic plants were found to take up more cobalt-60, almost 2.8 times higher amount of cobalt-60 compared to control plant, and they were really useful for a low-level nuclear waste which contains cobalt-60. This is a control plant. This is a transgenic plant which grows in cobalt-60. You can see the transformed plants becoming pale yellow in color after growing in cobalt-60 medium, whereas the control plants don't take up cobalt-60 and remain green. Then there are development of transgenic. After transgenic plant came this CRISPR-Cas9 or gene editing. This came only in the year 2013. And people have developed many plants with using the gene editing techniques. A corn plant with high amount of amylopectin, flax plant with enhanced omega-3 oil, rice, wheat, maize, tomato, potato, soybean with increased yield and mass, plants which are resistant to heat, resistant to cold, resistant to viruses, bacteria and fungal attack, and mushrooms resistant to browning. So many different types of plants were developed using the new techniques, that is genome editing, gene editing, or CRISPR-Cas9, for which the two ladies got a Nobel Prize last year. The plants for cleaning up of water bodies. Plants can be used not only for food and feed and medicines, but also for protecting our environment. Life on this planet is directly, all our life on this planet is, and even animals' life is dependent directly or indirectly on plants, because plants have to produce the food and then only they can have their food. So besides, plant release oxygen into the atmosphere, which is absolutely essential for life on the planet to survive. Without plants, we would not have survived at all, because we had no oxygen to breathe. We would not have any food to eat. So in 21st century, with increased industrialization, urbanization, and anthropogenic activities, the levels of pollutants have increased in the environment. So effective remediation methods are required for the cleanup. And at the moment, chemical and physical methods are used, but they're expensive and environmentally destructive. Alternate method is phytoremediation. What is phytoremediation? Is the use of plants to remediate pollutants from the environment. That is known as green technology. And this is a suitable method for developing countries because it's very cheap. There are several words like phytoremediation, phytoextraction. Plants can be used for extracting metals from soil and solutions. Rhizofiltration. Rhizofiltration is use of roots for filtration of dangerous chemical metals. And phytostabilization, suppose a particular area is contaminated with some contaminants, you can grow the plants, particular plants around it, and it can act as a biocurtain or a green curtain so that the pollutant does not come out of the particular place. Then some of the like arsenic and mercury and can be phytovolatilized into the atmosphere. So this is phytoremediation. It's aesthetically pleasing, economically cheaper, but it is very slow. Plants, as you know, have extensive root system. They are solar-driven pumps, and pollution are of two types: metals, metalloids, and radionuclides. And then second type of pollutants are organic pollutants. The difference between metals and organic pollutants are metals are contained, but they cannot be degraded, whereas organic pollutants or xenobiotic pollutants can be phytodegraded. Sometimes they can be mineralized. Two types of pollutants. One is organic pollutants. Organic pollutants, sometimes they're when it's man-made, it is called man-made also. And then it can be biodegradable or non-biodegradable, whereas inorganic pollutants like metals, non-metals, and radionuclides cannot be biodegraded. So phytodegradation of organic pollutants. There are a lot of organic pollutants in the atmosphere. A lot of herbicides people are using to control weeds like 2,4-D. Pesticides are thrown into the fields like chlorpyrifos to control insects. Lintane, trifluoralin. Then many of the companies are throwing chemicals into the water bodies. TCE, EDB, chloroform, carbon tetrachloride, etc. A lot of polyaromatic hydrocarbons from automobile industry are coming into the atmosphere to the soil and to the solution, like anthracene, benzanthracene, naphthalene, etc. Then explosives, lot of explosives like GTN, PTN, RDX, HMX. Then recently people are having a lot of drugs, and all these drugs also come out indirectly into the soil and solutions. So plants have got very extensive root system. The plant, in the soil, all these chemicals are there. They can take up the metals. They can take up the pollutants, and the metals are transported to the top, whereas non-metals like xenobiotic pollutants are not degraded. So metals and radionuclides include zinc, copper, cobalt, chromium, manganese, nickel, cadmium, then radionuclides like uranium, cesium, strontium, plutonium, etc. So what are hyperaccumulators? There are a group of plants which accumulate metals at a very high concentration in their shoot biomass, and they can be used as a real candidate plant, as a source for taking out the genes for a hyperaccumulation. They are low biomass plants. They are limited in geographical distribution. They are very small plants, so we cannot actually use it for cleaning up of our environment. So we have to develop candidate plants with potential to accumulate metals. So phytoremediation can be improved by transgenic technology. We have worked on a plant, miracle plant called vetiver ramachum, or a khas in Hindi. But this is Vetiveria sisanoid. The root system is about 8 to 10 feet. You can see a man standing here. You can see the extensive root system. When you grow this plant can control flood. If you grow the plant near the rivers, it can control the flood, and then it is a source of, it can tolerate high amount of alkalinity and acidity. And this plant can grow under different conditions, like grow under high temperature, low temperature, without water, with water, under flooding, high acidity, high alkalinity, high pH, low pH, etc. So we developed a technology for a large multiplication of this plant in vitro. And I'm just showing the slide how we tested this plant for uranium uptake. So this plant was tested for uranium uptake. Being in BRC, we wanted to see how this plant can remove effluents from our uranium plant in Jediguda, and also we grew the plant in a tailing soil. So the Bhabha Atomic Research Center, after extracting uranium, they have got a three big tailing ponds in Jediguda, which contain very low concentration of uranium. So this plant was kept as a biocurtain around the tailing ponds, and we studied the different parameters, and the plants could take up when the plant is grown in different concentrations of uranium, starting from 40 ppm to 300 ppm. Within 4 days, the plant could take up all the uranium, most of the uranium, up to 100%. Okay. And then you look at the plants which are growing in 25 ppm of uranium. They are very fresh, and the root system is also very good. Nothing happens to the root. They look very healthy. And we studied the growth of vetiver plants in places where in a uranium ore and in other solutions and the effluents from different chemical plants. So the plant could grow in uranium ore, and when you add citric acid, the plant could take up more uranium into the shoot biomass. We studied different pollutants. One is ruthenium nitrate. Then the plant, most of the plant, up to 20%, it goes very well. Then you can even extract uranium from these plants which were grown in uranium solutions. As you can see the yellow metal at the bottom of the flask, which is uranium extracted from the plant after growing in the thing. So 3 days after transplantation into the milk tailing, the plant is happily growing. Then after 82 days, it's growing very well. After 1.5 years also, the plant is growing. So when we wanted to see what happens to uranium at the end of the growth. So this is a plant which is exposed to 300 ppm of uranium, and this is a control plant. This is exposed to very high concentration of uranium, just like X-ray. This is a sort of X-ray, or we call auto radiograph. When you look at the auto radiograph, the uranium in the low concentration in the plant which was exposed to low concentration, uranium is accumulated. You can see the black spots in the root only, but when you grow in high concentration, it is translocated to the shoot. So this plant was grown in the uranium milk tailing ponds in Jedguda, and we can use this plant for sustainable environmental and bioengineering techniques. This plant was grown in by one of my friends, Dr. Pradeep from Calcutta. He has a company, Biosefra Foundation. He used this vetiver plant for cleaning up of eco restoration of Baligata canal restoration in Calcutta, and he could clean up the canal. This is the canal in which the plants are grown, and the banks of the canal are, he is protecting it with jute and other biomaterials. You can see here the bank of the canal, and plants are grown as a floating garden sort of. This is a water body for eco restoration before treatment. And after treatment, the plants are grown in a floating bodies and for removing the pollutants. This is a vetiver plant. This is a floating garden of biophytophonic solar aeration system. This is the Patoli Lake rejuvenation in Calcutta, in the Calcutta Metropolitan Development under KMDA. The plants are grown in the lake, and afterwards the lake becomes very beautiful, and people come for shopping in and around the lake, because otherwise it was a very dirty area. I have visited this lake, and now it is a very beautiful spot, and so many shops are there around, and the whole lake is cleaned up. And our Methi River in Mumbai, and how dirty it is. So recently, I think they have started, Mumbai Metropolitan Region Development, MMRDA, has started a program, and I think Pradeep is doing this eco restoration of the banks and cleaning up of Methi River. He just has started. I think Aditya Thackeray also came for the inauguration of a Methi River cleanup using these vetiver plants. This is Pradeep standing, with these plants. These are the small plants just transferred, and you can see people walking in the field. So when you read what are the new frontiers in biosciences, you can, those who want to take, I think 2 days back I read in paper, recently after this COVID-19 has struck us, there's an increase in demand for people, for students, for biology subjects. So anybody can actually work on a development of bioplastics, biosensors, bioprinting, etc. These are some of the latest techniques. Plants which will degrade pollutants is another area in which people can work. Then people have developed meat in the lab with using muscle cells of cows, using 3D technology. That is cheek without meat, even vegetarian meat the people have developed. Then you can do artificial photosynthesis to make food with you can produce a leaf to do artificial photosynthesis to produce your food. You can use nanotechnology based principle, just like in lotus leaf, to produce a waterproof fabric and paints. You can develop food that does not decay, and you can use new methods of bioenergy, the development of bioenergy. Now, because of this COVID vaccine, you know that some of the people who are producing COVID vaccines at the moment, like Bharat Serum, and that person is just a food scientist, I think, agricultural scientist. He is now making the best vaccine, the indigenous vaccine. So you know how important it is to study biotechnology. Thank you all. Thank you for your attention.
Ma'am, that was really nice, and it was so informative, and it was so much relevant to the present system. I'm so happy that you came. It's amazing. It was so lucid, and it was so understandable, and you explained it like a professor, so easily understandable. I mean, and this is open to everybody. We have a YouTube link, so it is open to everyone. So whoever comes and clicks onto the link and watch, it's open to all our Christ universities, all our 5 campuses. Thank you. I would request Nikhil to take charge and ask for questions, and Amrita to conclude with the proposition of the vote of thanks after the questions. Ma'am, please kindly excuse me. I have an interview to be conducted,
Okay,
and
no
I have
problem.
given time. Yeah,
Okay,
and so I would just take leave of you right now. And Amrita is she'll take charge, and Nikhil will continue if there's any questions. Thank you very much. And ma'am, we would want you to visit Lavasa as well, anytime when you have time, because Lavasa will require you. It's a very
Okay.
green place.
Okay.
Okay. Over to Nikhil and.
First of all, thank you so much. As a science student, that was really nice to listen to after a long time, going back and listening to biotechnology. So I must mention with a deep sense of gratitude for sharing with us such valuable information on plant and on feeding the world and cleaning up water bodies. So thank you, ma'am, for taking your time out of your busy schedule. Sadly, we don't have any questions. So Amrita, ma'am, I would request you to propose a vote of thanks.
Good afternoon, everyone. It is an honor to propose the vote of thanks today. So on behalf of School of Law, Christ deemed to be University Pune Lavasa, I'm humbled to thank Dr. Susan Eapen, former scientist at Bhabha Atomic Research Center, Mumbai, for this insightful lecture in the milestones of biotechnology and its contribution for social transformation. I'm also pleased to thank our Director, Reverend Father Dr. Josie P. George, and our Head of Department, Dr. Daisy Alexander, all my fellow faculty members, non-teaching staff members, students, and our IT department. Thank you for all your support. Thank you so much, ma'am. Thank you, everyone.
Bye, Sunil.
Bye, ma'am. Bye. Take care. We'll meet sometime.
Most of the students must have been from law and other commerce and other fields, not science students.
Yes, ma'am. But then I think it was the talk was very relevant, because last few days we were discussing about social transformation, and using technology for social transformation is something new for us. And I think that was really brilliant. I hope to continue our talks, ma'am. Maybe sometime later when you come to Lavasa, maybe we can have more discussions. Maybe we can create something new. So please convey regards to sir also.
Yeah. Okay.